Good day, everyone. Today we'll be discussing CRISPR-Cas9 and animal models in biomedical research, advancing disease understanding and therapeutics through gene editing technologies. This is brought to you by Castro, Jumarito, Losbañez, and Suaverdez. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a gene editing tool that allows scientists to precisely modify DNA in living organisms. It was adapted from a natural system found in bacteria. So, how does it work? When bacteria are infected by a virus, they store a piece of the viral DNA as a genetic memory. If the virus returns, CRISPR uses a guide RNA to recognize and destroy it. In gene editing, scientists use a synthetic version of this system. A guide RNA directs the Cas9 enzyme to a specific DNA sequence. Now let's move on to the history of CRISPR. In 1987, Yoshizumi Ishino discovered unusual repeating DNA sequence in E. coli, marking the first observation of CRISPR. By 2002, Francisco Mojica named these sequences CRISPR after finding them in other microbes. In 2005, Bolotin and Haft linked CRISPR to bacterial immunity and identified Cas genes. In 2007, Baranjo and Horvat confirmed CRISPR protects bacteria from viruses. Between 2008 and 2011, researchers clarified that RNA guides CRISPR and proposed its classification. In 2012, Emmanuel Charpentier and Jennifer Doudna repurposed CRISPR-Cas9 as a gene editing tool, leading to their Nobel Prize in Chemistry in 2020. By 2013, CRISPR-Cas9 was used to edit human and animal genomes, launching its role in biomedicine.
The next thing that we will be talking about are the medical applications in relation to our study. In our study, CRISPR-Cas9 is highlighted as a transformative tool for generating precise genetic modifications in animal models. Its ability to introduce targeted gene edits has greatly improved our capacity to mimic human disease conditions, making it invaluable for studying pathophysiology and accelerating the development of potential treatments. Beyond its role in creating animal disease models, CRISPR has direct medical applications, such as treating genetic disorders, editing immune cells for cancer therapy, and so on. So, these advances, many of which were first validated in CRISPR edited animal models, demonstrate the translational power of this technology in clinical medicine. The first that we have are genetic disorders. An example of this is the sickle cell disease or the beta thalassemia. So CRISPR is used to edit blood stem cells by disabling the BCL11A gene, reactivating fetal hemoglobin to compensate for the defective adult form. Now these edited cells are returned to the patient, offering a long-term cure. So an animal model relevance for this is mice engineered with human blood gene mutations that were used to test this strategy's safety and effectiveness before moving to human trials. Moving on with cancer therapy. Immune cells like T cells are genetically modified using CRISPR to improve their ability to recognize and kill cancer cells. For example, CRISPR can remove PD-1 receptors or insert chimeric antigen receptors to enhance anti-tumor responses. So mouse models of leukemia, melanoma, and solid tumors are critical for testing the function and targeting ability of edited T cells before clinical applications. Next, we have our viral infections. So, one example of this is the HIV or the HPV. So, CRISPR targets and cuts out viral DNA from host genomes. In HIV, it aims to remove integrated proviral DNA, and in HBV, it disrupts viral replication by editing viral genes or cccDNA in infected liver cells. So, mouse models with humanized immune systems and livers are used to simulate persistent infections, allowing researchers to test delivery and off target effects.
Different mutations on the cystic fibrosis transmembrane conductance regulator, or CFTR, is what often leads to cystic fibrosis. These mutations affect the lungs, among other organs like that of pancreas, liver, etc. It causes the dysregulation of different ion transports that ultimately form mucus accumulations and impaired clearance in airways. But what are animal model relevance? Well, mice are typically what is utilized for cystic fibrosis research and have their own mouse lines that are readily available for such. With new technology, there are even mice that have strains of human cystic fibrosis causing mutations by gene editing them in. Sheep models have also been recently used as CRISPR can knock out their CTFR gene and be studied. When the pancreas are unable to produce insulin or it is ineffectively used by the body, the problem of diabetes arises as the body will not be able to regulate blood glucose. Type 1 diabetes is associated with deficient production of insulin, while type 2 is that the body cannot properly utilize said insulin. For animal model relevance, the leptin and leptin receptors were knocked out using CRISPR to study obesity and diabetes in mice to induce a mutation that showed phenotypic disorder highly identical to that of obesity and diabetes. While undoubtedly CRISPR has been a cornerstone in scientific progress, many still raise ethical concerns on the use of it. Animal research is guided by laws, public opinion, and moral views. The first and foremost concern is the welfare of animals being used in the laboratory or research settings with a need to reduce the number of animals in the lab, minimize pain and distress caused, and ultimately the need to seek out non animal related alternatives when possible. The progress of scientific research, after all, relies not only on what we can do, but how we do it.
As for the mechanism, CRISPR-Cas9 is a gene editing system adapted from bacteria. It uses a guide RNA to direct the Cas9 enzyme to a specific DNA sequence. Once the target is found, Cas9 creates a double-strand break in the DNA. The cell then repairs the break through one of two pathways, either non-homologous end joining, known as NHEJ, which is quick but error-prone, often causing gene disruptions, and homology-directed repair, or HDR, which uses a DNA template for precise edits. As for delivery methods, getting CRISPR into cells is a key step in gene editing. Common delivery methods include viral vectors like AAV or lentivirus, which is very efficient but may integrate into the genome. Next is lipid nanoparticles, also known as LNPs, which is non-viral and safer, especially for in vivo use. Next is electroporation, which uses electric pulses to open cell membranes. And lastly is microinjection, which directs injection into embryos or cells for precision. The delivery method depends on the target tissue, safety needs, and application. Next is the challenges and limitations. Despite its power, CRISPR faces several hurdles. First, off target effects, as Cas9 can cut unintended sites, leading to unwanted mutations. Next is homology directed repair inefficiency, as precise editing via HDR is limited and less efficient than NHEJ or non homologous end joining. Next is immune reactions, as some people may have pre existing immunity to Cas9 proteins. And lastly, ethical concerns, especially with the germline editing and unequal access to the technology. Now let's move on to CRISPR and how it is related to the chosen study. First, the mechanism transgenic model creation via CRISPR-Cas9. Researchers use Cas9 or guide RNA and a donor DNA template to introduce specific gene alterations, such as single nucleotide polymorphisms, into animal genomes to recreate human disease traits. For example, mice have been engineered to model cancer development by targeting genes like CRAS, TP53, and LKB1 using AAV or lentivirus mediated CRISPR delivery. As for delivery methods, as mentioned, viral vector mediated editing in vivo and ex vivo. The study describes using both AAV and lentivirus systems to deliver CRISPR components effectively into mouse neurons and endothelial cells. This approach enabled precise induction of mutations that model lung and liver cancers in mice. Lastly, the challenges and limitations. Model selection and translational relevance. Choosing the best animal model is complex. Studies point out that inappropriate model selection may yield misleading results or inefficient use of resources, especially if the physiology or disease progression doesn't match human conditions. Also, ethical considerations. As animal experiments face ethical constraints, notably regarding pain, distress, and welfare. Research practice is guided by the principles of the three R's: reduction, refinement, and replacement to minimize animal use and harm.
Now let's move on to the future prospects of CRISPR. In the future, CRISPR may go beyond editing DNA sequences to controlling how genes are expressed. One promising area is epigenetic editing, which adjusts gene activity without changing the DNA itself and may offer safer, reversible therapies. In agriculture, CRISPR is being used to create crops that can resist pests and survive drought, helping them adapt to climate change. Researchers are also using artificial intelligence to improve CRISPR tools by predicting outcomes, avoiding errors, and designing better enzymes and guide RNAs for even more precise editing. Thank you
Here
all
are
for
our
listening.
references. Thank you and have a great day.